Okay, let's continue. We finished up in our last video talking about the neuromuscular junction. If you haven't watched it, please go back and watch it. But now let's talk about the functional unit of muscle. Okay, the functional unit of muscle is called a sarcomere. It's got a lot of different moving parts here, but I just want you to see this picture and then we'll start breaking it down. So remember before we talked about action potentials. We said the neurotransmitter acetylcholine is stored in these vesicles. Calcium comes in to the axon terminal, tells the neurotransmitter acetylcholine in these vesicles to open up. So acetylcholine is released in the synaptic cleft. It's going to bind to the acetylcholine receptor on these sodium channels. The sodium channel opens and sodium rushes in, creating an action potential. Now, this structure here, this invagination right here, is called a transverse tubule or a T-tubule. So this T-tubule or transverse tubule is this structure right here because it runs transversely to everything else going this way. This is another transverse tubule. The transverse tubules contact a structure called the SR or the sarcoplasmic reticulum. That's this blue meshwork here. Now, the sarcoplasmic reticulum is a really, really unique structure. It stores a mineral. What mineral do you think the sarcoplasmic reticulum is going to store is involved with muscle contraction? Calcium. It's going to store calcium. So when the excitation, depolarization comes down through the transverse tubule, it's going to migrate, move inward to the sarcoplasmic reticulum, telling the sarcoplasmic reticulum to release calcium. And calcium is going to have a major, major impact. It is a major player in terms of making muscles shorten and lengthen to contract. Okay? I also want you to see that there's lots of mitochondria here. This is a mitochondria, mitochondria. Muscles have lots of mitochondria because mitochondria are going to produce energy in the form of ATP. ATP is adenosine triphosphate. Okay, so let's review. Let's see what we have here. Here is a somatic motor neuron. Here are the dendrites. This is the cell body. Here's an axon. The axon looks like it's myelinated by the Schwann cell that produce myelin, that fatty cholesterol layer to protect nerves. And at the very end are the axon terminals with the end bulbs storing that neurotransmitter. So right here in this box, this is the neuromuscular junction. Let's see what's happening here. So this part here would be the very end of the axon, the end bulb with these neurotransmitters. This would be the synaptic cleft, and then this would be the motor side or the muscle side. So this neurotransmitter acetylcholine is going to move into the synaptic cleft when calcium tells it to open up through excitosis. Sodium channels open, sodium rushes in and creates this excitation, right? This is the depolarization, excitation, action potential. This is the T-tubule, that invagination that we saw here, these invaginations. That's the transverse tubule, which is here. And we know the transverse tubule or the T-tubule butts up against the sarcoplasmic reticulum. The blue structure stores calcium. So here is the invagination, the T-tubule butts up, touching and making contact with the sarcoplasmic reticulum, which stores calcium. So it's going to tell calcium to be released. The calcium is going to be released, and it's going to hit this structure here, which is your thick and thin filament called actin and myosin. Myosin, the thicker filament that has these heads, the myosin heads, actin is thin. Actin is thin. Somehow, this calcium is going to be involved in making a muscle contract. And that's what we have to get into next. So to understand how calcium is involved with muscle contraction, we have to first understand that myosin and actin are the contractile proteins, and the regulatory proteins, the on-off switch, are troponin and tropomyosin. Troponin and tropomyosin. So what we see here on the left, this is myosin, and what we see here, the yellow circular spheres, that's actin. We can see here actin. So you have actin and myosin. Now, what I want to see here is that each of these yellow spheres or circles or pearls, these are G molecules, like individual G molecules, call them. On each G molecule, you can see that there's a little black dot, target, or circle on each of these spheres, but you don't see the entire circle. Here they call it a myosin binding site. The myosin binding sites are covered. They're blocked or shielded by this rope called tropomyosin, this long string. And here's another string here intertwining. Both of these strings or ropes are called tropomyosin. It's another type of protein. It's a regulatory protein. And then these three little blue circles here, there's another regulatory protein called tropin. So in my mind, when I teach this to my students, I like to keep it simple. I try and tell my students, think of the troponin as like this long rope protein, a string, and it's blocking these binding sites, but we need something to hold it in place, so we need a staple to hold down the tropomyosin. The staple is troponin. So troponin is a staple that's holding down a string. All of these are staples, staple, staple, staple. The staples are troponin, the string of rope is tropomyosin. Okay, here is the myosin and the myosin heads. So here at the end of the myosin head, it says this target right here is an actin binding site. So somehow myosin and actin need to connect. They need to make contact. So this is a myosin head. Here, my fingers and my fist, and this actin, this is myosin. I have to find this binding site. But here, the binding sites are blocked. Okay, keep that in mind. In order to unblock it, we need to remove the staples. We need like staple remover. If you can remove the staples, if you can remove troponin, then tropomyosin can shift, move out of the way. And if tropomyosin shifts out of the way, this binding site on actin becomes opened and exposed, so that the myosin heads can find a place to contact and create the contraction. Okay, so these are really important. Now, what do you think is going to be the staple remover? It is a mineral called calcium. So calcium is involved in removing these troponin or the staple so that the tropomyosin shifts. I'm going to go over it again with you. These structural proteins are titin, nebulin, myosin, dystrophin. These are structural proteins that give muscle the direction of fibers to maintain its structural components. There's a condition called muscular dystrophy, in which the body breaks down some of these proteins. Okay, so we have these myofibrils that are made up of thin and thick filaments, actin and myosin. They're arranged in these contractile units called a sarcomere. Sarcomere has the two regulatory proteins that act as an on and off switch, tropomyosin and troponin. We have the contractile proteins, actin and myosin. Actin is thin, myosin is thick. Then the structural proteins that I mentioned before that deal with the alignment of the muscle, its extensibility, elasticity, and stabilization. When we talk about actin, we have the F and G's, right? The G are those G molecules, which has the binding site. So we have the G molecules, tropomyosin, and troponin. The tropomyosin, I said, is a rope-like structure that covers the entire length and covers the myosin binding sites. Tropomyosin is on actin. The troponin, I said, think of it as the staple that's going to hold the tropomyosin over those G proteins or those G molecules. Now, what is the staple remover? We think of calcium as the key to the lock. It's going to remove the troponin, so tropomyosin shifts, the binding sites are exposed, and actin can make its contact. So here, you see each of these is a G molecule. This is F strand. This is another F strand. So all the G come together to make an F strand. And you can see that each G molecule of actin, you can see in this case, purple, 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 all those target sites. These are the binding sites. Can't fully see them. They're not exposed because the tropomyosin is blocking it, and the troponin, which is the staple, is holding it in place so that the myosin head can't bind to create the contraction. These are the myosin heads. When you think of myosin, think of it as like your forearm and elbow. So you have your elbow has a bend to it, and then you have your wrist that has a bend to it. So at first, your elbow comes up, the myosin head is on top. It can attract to the binding site on actin, and when it does, then the wrist flicks and creates this slide or the sliding filament theory of making a muscle contract. So myosin bends here, makes contact, and then it bends here, creating the movement of actin. Okay. So you can see down here on the bottom, you can see here's the myosin tail and here's the head. Here is a hinge and here's a hinge. So this would be like the elbow, the wrist, and this is the myosin head that can pivot and flick. Up here is another bunch of G molecules with the binding site. You could see tropomyosin blocking, and you could see troponin holding the tropomyosin in place. On the top, we see a sarcomere at rest. If you look at the H zone right here, it's nice and wide. Here you can see the H zone shortened. So this is in a contracted state. That means that the myosin heads found a way to bind to actin and bring them closer together. Okay, I'm going to take a break here, and when I come back, we'll go through once again and review the different phases of how the neuromuscular junction works, about calcium, and about the contraction that takes place. In fact, you know what? Let's finish up with this. I don't need to take a break here. Let's finish this section here because it's a little bit of an overview. So you can see right here is the neuromuscular junction. Here's the somatic motor neuron. Here's the synaptic terminal. These little red circles, once again, the neurotransmitters. Here's the synaptic cleft. We remember that calcium comes in, tells the neurotransmitter called acetylcholine to release its contents through exocytosis in the synaptic cleft. It's going to bind to the acetylcholine receptor or the sodium channel. Sodium channel opens, and you see the excitation goes down the transverse tubule, that invagination. The nerve impulse or action potential connects this sarcoplasmic reticulum. Remember, the SR stores calcium. It tells the sarcoplasmic reticulum to release calcium. Calcium is released. Now it's going to act on the sarcomere where you have actin and myosin. Remember, actin had the regulatory proteins, troponin and tropomyosin. Tropomyosin blocks the binding site, but what holds it in place is troponin. So the calcium that is being released right here is remove troponin, so tropomyosin shift, and now myosin, you have one hinge here, one hinge here. So this hinge bends, now the myosin binds to the binding site of actin, and after that elbow bends and makes contact, then the wrist flicks and creates a shortening contraction that we see down here at number 5. Now, to tell us to go back to its resting state, you just got to reverse steps, right? So we got to get rid of, to end the contraction, you got to remove neurotransmitter. So acetylcholine is removed by acetylcholine esterase. Anything that ends in ACE is an enzyme. Enzymes degrade things and break things down. So acetylcholine is removed by acetylcholine esterase. When there's no more neurotransmitter, calcium goes back into the sarcoplasmic reticulum. If there's no more calcium, then troponin and tropomyosin can go back to their resting state. The myosin head goes back, and the muscle contraction ends. Now, in order to do that, just understand the steps that initiate the contraction. The key steps at the neuromuscular junction, we talked about acetylcholine that is being released. We talked about the change in the action potential going down the transverse tubule. The action potential binds and connects to the sarcoplasmic reticulum that stores the calcium. It releases the calcium, and when that calcium hits the sarcomere, where you have actin and myosin, the calcium is going to bind to troponin, the staple, remove it. Tropomyosin complex shifts, the orientation of the tropomyosin is going to move, exposing the binding site, unactive. And then the cross bridge forms, meaning the myosin head binds to actin. They call that a cross bridge. Okay, then the contraction can begin. The contraction begins because this slide happens, but then the myosin head needs to detach in order to grab on again and contract. And then the myosin head has to detach only to bind again and contract. Think of it as like a tug of war where I'm holding a rope, I pull the rope and I can't pull anymore. I have to release, grab the rope again and pull. Each time my hand releases, that requires energy. So keep that in mind. The detachment is powered by hydrolysis of ATP. It takes energy, or ATP, to release the myosin heads, which explains why when you see a person that's in rigor mortis when they die and they're in this shortened state, right, the muscles are not in a relaxed state. They're in a state of rigor mortis where the body is in a shortened, contracted state, because if you're dead, you're not making energy, or ATP, to release the myosin heads from the active. Okay? In order to end the contraction, that's where the acetylcholine esterase breaks down the neurotransmitter, and then all the calcium is reabsorbed back in the SR, or the sarcoplasmic reticulum. If there's no more calcium, then troponin and tropomyosin go back, they block the binding site, and there's no more cross bridge, and then the muscles are relaxed. Okay, so let's end this video here.